Take a drop of mud from the bottom of a coastal pond, smear it thin between two pieces of glass, and put it under a microscope. What you get is chaos. Thousands of bacteria moving in every direction at once, bumping, spinning, reversing, going nowhere in particular. That's what bacteria in a water sample look like. That's what they've always looked like. Now bring a small bar magnet up to the edge of the slide. Within a few seconds, a crowd forms. It's fast, and it's obvious even at low magnification. A hard, visible migration of cells toward one specific edge of the drop, packing themselves against the glass in a dense band you can see with a cheap lens. Turn the magnet around, and the entire crowd reverses. Every cell in the band swings its body 180 degrees and swims back across the field of view to the opposite edge. You can do this over and over. They will follow the magnet until they run out of energy. Richard Blakemore was a graduate student at the University of Massachusetts when he watched this happen in 1975, working with mud from New England coastal sediments. He wasn't looking for it. He was looking at microbes in pond sediment, saw a population of cells that kept ending up on the same side of the drop no matter how he prepared the slide, and eventually worked out that the side they favored was north. He published the result in Science that year under a title with no drama in it at all: Magnetotactic Bacteria. 2 words, 3 pages. He was not actually the first. A physician named Salvatore Bellini, working in Pavia, Italy, had seen the same behavior in fresh water samples around 1963, more than a decade earlier. He wrote it up in Italian in internal reports from his institute and called the cells magnetosensitive. Almost nobody outside Italy read them. Those reports sat unnoticed until the 2000s, when they were translated and circulated, and Bellini got his credit posthumously, which is its own small tragedy and also completely typical of how science actually works. But it isn't the interesting part. The interesting part is what's inside those cells. Because when Blakemore and Richard Frankel put the bacteria under an electron microscope, they found the reason. Running down the long axis of each cell, roughly through its center, was a straight line of dark particles. Between about 15 and 30 of them in a typical cell, though the count varies by species, each one a crystal. Each crystal wrapped in its own membrane sac, each sac clipped into position so the whole line held straight, like beads strung on a wire that refuses to bend. The crystals were magnetite, iron oxide, the same mineral that makes lodestone magnetic, the stuff that got dragged across the floor of the ancient world on a string and pointed north and started the whole business of navigation in the first place. And here it was, grown inside a single-celled organism, in a compartment the cell had built for the purpose, at a size the cell had chosen, in a chain the cell had assembled and anchored. A compass. Manufactured from scratch, from dissolved iron, by something with no brain, no eyes, and a genome smaller than a rounding error. That is the story I want to lay out here, and I want to be exact about why. Because the question of how a living thing builds a magnetic sense inside its own body has been open for about 50 years, and it has produced an enormous amount of very good behavioral science, a fair amount of very bad cell biology, at least 3 major retractions of interpretation, and 1 solved case, exactly 1, in 1 organism. And that organism is a bacterium you can pull out of harbor sediment with a magnet and a plastic syringe. Everywhere else, in every animal that has ever been tested and shown to use the geomagnetic field, we have behavior without hardware. We know sea turtles read the field. We know salmon read it. We know bonnethead sharks read it, and spiny lobsters read it, and eels read it. And every year the behavioral evidence gets tighter and better controlled. What we cannot do is point at a cell in any one of those animals and say, there, that's the receptor. That's the crystal. That's the wire it's attached to. So the honest way into this is backwards. Start with the case that closed. Take it apart piece by piece all the way down to the genes, because in the bacterium we really do have it down to the genes. And that gives us something almost nobody has when they argue about animal magnetoreception, a specification, a real one. A parts list for a working biological compass, written by evolution, verified in the lab, and reproducible on demand. Then take that specification into the ocean and go looking. Let me set the scale first, because the numbers here are strange and they matter for everything that follows. Earth's magnetic field at the surface runs somewhere between about 25 and 65 microtesla, depending on where you're standing. Weakest near the magnetic equator, strongest near the poles. To give that a physical meaning, a cheap refrigerator magnet is roughly 100 times stronger at its surface. A hospital scanner is something like 60,000 times stronger. The field a sea turtle navigates an entire ocean basin with is, by any laboratory standard, almost nothing. It's a whisper. And the problem with detecting a whisper inside a living body is not amplification, it's noise. Everything inside a cell is being shaken constantly. Water molecules at body temperature are moving at hundreds of meters per second, colliding with anything suspended in them millions of times a second, and that random battering has an energy scale. Physicists call it KIT, the thermal energy. And at the temperature of seawater or blood, it comes out to about 4 times 10 to the minus 21 joules. Very small in absolute terms, but it's the floor. Any structure in a cell that wants to hold a position, or register a force, or keep an orientation, has to beat that floor. If the energy your sensor gets from the magnetic field is smaller than the energy the water is dumping into it at random, your sensor is a thermometer. It's measuring the temperature. It just doesn't know it. This single constraint is the reason animal magnetoreception has been so brutally hard, and it's the reason the bacterium is such a beautiful piece of engineering. Because the bacterium solves the noise problem in the most direct way available. It builds a permanent magnet big enough that the field wins, not much bigger. That's the part that gets me. It doesn't overbuild. It builds a magnet that beats thermal noise by roughly an order of magnitude and then stops, because going further would cost iron and energy for no gain, and because past a certain size the physics turns on you and the magnet stops working. There's a window, a narrow one, and the cell hits it, every time, across species, across continents, in crystals grown independently in organisms that split from each other a very long time ago. Here's the part that I think gets undersold every time this story gets told. Making magnetite is not hard. Iron oxide precipitates out of solution in all sorts of ordinary chemistry. Rust is easy. Sediment is full of magnetic minerals that formed with no help from biology at all, from volcanic rock breaking down, from lightning strikes, from fires. If all a cell needed was some magnetic material inside it, that would be a solved problem the day life invented iron metabolism. What's hard is control, building crystals of a specified mineral phase, in a specified size range measured in tens of nanometers, with a specified crystal habit, at a specified location in the cell, arranged in a specified geometry, anchored against forces that want to pull them into a useless clump, and doing it reproducibly in every daughter cell for as long as the lineage lasts. That's manufacturing. That's a production line. And the reason we can talk about it as a production line is that over about the last 30 years, a set of labs in Germany, the United States, Japan, France, and China took the thing apart gene by gene, deleted each part, watched what broke, and put it back together. They know which gene makes the compartment. They know which gene pulls the iron in. They know which gene decides how big the crystal gets. They know which gene builds the rail the crystals sit on, and which gene glues them to it, and what happens to the cell when you cut that glue. And in 2014, a group led by Isabel Kalinko in Dirk Schuller's lab did the thing that closes a case in molecular biology. They took the relevant gene clusters out of a magnetic bacterium, moved them into a completely different bacterium that had never made a magnetic crystal in its evolutionary history, a purple photosynthetic organism called Rhodospirillum rubrum, and switched them on. The recipient built magnetosomes. Not perfectly, not as many, and the crystals came out somewhat different. But it built membrane compartments, filled them with magnetite, and lined them up in a chain. A bacterium that had never done this before was handed roughly 30 genes and started manufacturing compasses. That result was published in Nature Nanotechnology, and it means the parts list is not just correlated with the ability. It's sufficient for it. Hand over the genes, get the organelle. Nobody has ever done anything remotely like that with an animal magnetic sense. Nobody is close. Nobody knows which genes they would move. So that's the shape of what's in front of us. On one side, a system understood well enough to transplant. On the other side, animals that have been crossing oceans on magnetic information since before there were oceans shaped like the ones we have now, and a search for their receptor that has been running for half a century and has produced, so far, a series of very instructive mistakes. Let's go inside the cell. The first thing the cell builds is an empty bag. That's worth sitting on for a second, because it's the opposite of how most people imagine this working. The intuitive picture is that iron accumulates somewhere in the cytoplasm, crystallizes, and then the cell tidies up around it, wraps it, deals with it after the fact. That's backwards. The compartment comes first. The bag is built, positioned, and stocked with a specific set of proteins before there is any iron in it at all. And you can prove that in the crudest possible way: starve the bacteria of iron. Grow them in a medium where there's almost nothing for them to work with. Then look at them under a cryo-electron microscope. The bags are there. Rows of them. Empty membrane compartments lined up along the axis of the cell exactly where the crystals would normally sit, containing nothing. Feed those same cells iron, and within roughly 15 to 30 minutes, crystals begin appearing inside the existing compartments. The factory floor was already built. It was waiting for stock. What are those compartments made of? For a long time, the honest answer was that nobody was sure. There were two possibilities on the table. Either the magnetosome membrane was a free-floating vesicle, budded off and released into the cytoplasm the way a eukaryotic cell buds vesicles off the Golgi, or it was still attached to the cell's inner membrane, an invagination, a pocket pushed inward, like a finger poked into the side of a balloon. This got settled in 2006, and the way it got settled is a nice demonstration of how much of biology is really a technology problem. Arash Komeli, working with Grant Jensen at Caltech and colleagues, used electron cryotomography on whole intact cells of Magnetospirillum magneticum, strain AMB1. The technique matters. Conventional electron microscopy requires you to chemically fix the sample, embed it in resin, and cut it into slices thinner than the thing you're trying to look at. You get beautiful images of something that is, by then, a chemically modified corpse arranged into slices. Cryotomography freezes the cell so fast that the water inside it doesn't have time to form ice crystals. It goes into a glassy state. The cell is preserved in something close to its living arrangement, and then you tilt it in the beam and reconstruct it in 3 dimensions. When they did that, the magnetosome membranes showed up as invaginations, pockets of the inner membrane pushed inward, in many cases still connected to it by a narrow neck. Some of the compartments in a chain were open to the outside membrane, others appeared pinched off. The result was published in Science, and it did 2 things at once. It answered the topology question, and it embarrassed a textbook. Because for most of the 20th century, the clean dividing line between bacteria and everything else was compartments. Eukaryotes have organelles. Bacteria are a bag of enzymes. That was the line you learned. It was always a little bit of a lie, and the magnetosome is one of the sharper knives in the pile that killed it. Here's a bacterium with a membrane-bound organelle, with its own protein composition, its own chemistry, its own position in the cell, and its own inheritance. That protein composition is the second surprise. If the magnetosome membrane were simply a piece of the inner membrane folded inward, you'd expect it to have the same proteins as the inner membrane. It doesn't. When you purify magnetosomes and run their membranes on a gel, you get a distinctive set. Something on the order of 20 to 30 proteins are strongly enriched there compared to the rest of the cell's membrane, and some of them are found essentially nowhere else. The cell isn't just folding membrane inward, it's sorting. It's deciding what gets to be in that pocket and what stays outside. That's a lot of specificity for a bag, and it tells you the bag is doing chemistry. So why bother? Why build a room at all? Why not precipitate the crystal in the open cytoplasm and save yourself the trouble? Two reasons, and both are severe. The first is that iron in solution is dangerous. Free ferrous iron in the presence of hydrogen peroxide runs a reaction chemists call Fenton chemistry, and the product is the hydroxyl radical, which is about as aggressive an oxidant as biology ever has to deal with. It attacks DNA. It attacks lipids. It attacks proteins more or less indiscriminately. Every organism on Earth that uses iron, which is essentially all of them, has evolved elaborate machinery for making sure iron is never just sitting around loose. It's bound to transport proteins, chaperoned, locked inside ferritin cages, handed off directly from one carrier to the next. Loose iron is a fire hazard, and to grow a magnetite crystal, you need iron concentrations far above what a cell can safely tolerate in its cytoplasm. You need local supersaturation. The only way to have supersaturation in one place and safe conditions everywhere else is a wall. The second reason is finer, and it's the one that actually determines whether you get magnetite or something useless. Magnetite is a mixed valence mineral. Its formula contains iron in two different oxidation states, ferrous and ferric, in a fixed ratio of 1 to 2. That ratio is not optional. Get it wrong, and you don't get magnetite. You get one of the other iron oxides, and there are a lot of them, and almost all of them are magnetically hopeless. Fully oxidized iron gives you hematite or goethite, which are antiferromagnetic and essentially useless as a compass. Push too far the other way, and you get other phases with other problems. The window in which magnetite is the stable product is defined by pH and by redox potential, and it is not especially wide. So the cell has to hold a specific pH and a specific redox potential inside the compartment, different from the cytoplasm around it, for as long as the crystal is growing. You cannot do that in an open room. You need a membrane, and you need proteins in that membrane that pump, that transport, and that shuttle electrons. That's what the magnetosome membrane is for. It's a reaction vessel with its own atmosphere. The genetics behind making the vessel itself have been narrowed down with the usual brutal method. Delete a gene, see if the cell can still do it. In AMB1, work from Comeli's lab identified a small core, set required for the membrane compartments to form at all. Take out any one of a handful of genes, including the ones called MAM-I, MAM-L, MAM-Q, and MAM-B, and the cell stops making magnetosome membranes entirely. No compartments. No empty bags. The count goes to 0. Put the gene back and the compartments return. That's a clean result. And it's the kind of result that's essentially impossible to obtain in an animal, for reasons we'll get into later, because you can't casually knock out candidate genes in a loggerhead turtle and check whether it can still find Cape Hatteras. Two of those proteins are worth naming individually because they show up again. MAM-B and its close relative MAM-M belong to a protein family called cation diffusion facilitators. This is an ancient, widespread family whose day job across most of life is getting toxic metal ions out of the cytoplasm, usually zinc, sometimes iron, sometimes cobalt. They're pumps. In the magnetosome, they've been repurposed to move iron across the magnetosome membrane and into the compartment. So the transporter that delivers the raw material to the crystal is a modified version of a protein whose original function was probably getting rid of metal, running in what amounts to a controlled direction. That kind of thing shows up constantly once you start looking at how new biological machines get made. Evolution has no design office. It has a scrapyard. MAM-L is a small, unusual protein with a stretch of positively charged amino acids at one end, and it looks like it's involved in the physical act of bending the membrane inward. Bending a membrane is not trivial. Eukaryotic cells have entire protein families dedicated to it, coat proteins that assemble into curved lattices and deform the membrane by brute geometric force. Bacteria don't have those. Whatever the magnetotactic bacteria are doing to invaginate their inner membrane, they're doing it with a small, idiosyncratic toolkit that appears to have been assembled for this specific purpose and doesn't have obvious relatives elsewhere. I find this more impressive than the crystal, honestly, and the crystal is the famous part. The crystal is chemistry. And chemistry, given the right conditions, is inevitable. Deciding where in the cell to build a room, building it out of a membrane that doesn't want to bend, and stocking it with a curated set of proteins that will hold a chemical environment nothing else in the cell is holding. That's the hard engineering. The mineral is what happens after the hard part. So here's the state of the factory at this point. A row of membrane compartments, invaginated from the inner membrane, positioned along the long axis of the cell. Each one carrying a specific protein set including at least 2 iron transporters, each one capable of holding an internal chemistry distinct from the cytoplasm. All of it built before any iron arrives. Now the cell has to get the iron in, decide what to do with it, and stop at exactly the right moment. If you sequence the genome of Magnetospirillum gryphoswaldense, the German workhorse strain that most of this research runs on, and you go looking for the genes that make magnetosomes, you find something you would not expect. They're all in one place. You'd expect them scattered across the chromosome, the way genes for a complicated trait usually are. They aren't. They're clustered. A single contiguous stretch of DNA, roughly 100,000 base pairs long in that strain, sitting in the genome like a separate document that got pasted into a larger one. Researchers call it the magnetosome island, and the name is doing real work because the region has all the standard fingerprints of a genetic module that arrived from somewhere else. Its base composition is different from the rest of the chromosome. Its edges are littered with the mobile genetic elements that cut and paste DNA around. Transposase genes, insertion sequences, the molecular equivalent of packing tape. And it's unstable. That's the detail that gets me. If you grow these bacteria in the lab for long enough, without ever selecting for magnetism, without ever using a magnet to pull out the cells that respond, a fraction of the population spontaneously loses the ability. They don't lose it by accumulating point mutations. They delete the island, whole chunks of it, tens of thousands of base pairs at a time, dropped out of the genome under stress. There are well-characterized lab strains that arose exactly this way, non-magnetic derivatives of magnetic parents, missing a large block of that region. Think about what that implies. The compass is on a cartridge. It's a module the organism can gain and can lose, more or less as a unit, which is a strange way to carry an ability that seems so central to how the organism lives. And it's also, I suspect, exactly why the ability shows up in bacterial groups that are only distantly related to each other. This module travels. Inside the island, the working genes are organized into 4 main operons, which is to say 4 bundles of genes that get switched on together. The biggest one is called MOM-AB, and it carries something like 17 genes. This is the core. Delete the MAM-AB operon. and you get nothing at all. No compartments, no crystals, no chain. Put MAM-AB back, by itself, into a strain that's lost the whole island, and the cell starts making magnetosomes again. Small ones, irregular ones, fewer than normal, and with sloppy crystal habit. But real magnetosome membranes with real magnetite inside them, arranged in a rough chain. So MAM-AB is the machine. Everything else is quality control. The second bundle is called MAM-GFDC, 4 genes, and the proteins they encode sit in the magnetosome membrane in large amounts. Delete all 4 and the cell still makes magnetosomes, still makes chains, still swims toward a magnet. The crystals just come out too small. In the published work on this from Dirk Schuler's group, deleting the whole MAM-GFDC set dropped average crystal size to roughly 3/4 of normal. And here's the part that says something about how the system is built. Putting back any single one of those 4 genes partially restored the size. They're partly interchangeable. It's a dial with 4 hands on it, and the position of the dial depends on how much total protein is present rather than which specific one. Third bundle, MMS6. The protein it encodes is small, acidic, and it binds tight to the surface of the growing crystal. Tight enough that when researchers purify magnetosome crystals and strip everything off them, MMS6 is one of the proteins that keeps clinging on. Take the gene out, and the crystals come out smaller and structurally messier. Put purified MMS6 protein into a test tube with iron solutions and no cell at all. And it changes the size and shape of the magnetite that forms. That's an unusual level of evidence for a single protein's role. It works outside the organism. Fourth bundle, MAM-XY, which handles crystal maturation and redox chemistry, and which includes a gene called MAM-Y that turns out to be involved in constraining the diameter of the magnetosome compartment itself and helping position the chain along the cell's long axis. Add all that together, and you get a parts list of roughly 30 genes doing the essential work. 30. That's a small number for a machine this specific. For comparison, a bacterial flagellum, the rotary motor that lets these same cells swim, takes something in the range of 40 to 50 genes. Building a compass is roughly as genetically expensive as building a propeller. Now, the experiment that turns all of that from a strong inference into a demonstration. In 2014, Isabel Kalinko and colleagues in Schuler's lab took those 4 operons, about 30 kilobases of DNA in total, and moved them into Rhodospirillum rubrum. That's a purple non-sulfur photosynthetic bacterium. It's a well-studied organism. It's used in labs all over the world, and it has never in its evolutionary history made a magnetic crystal. It has no magnetosome island. It has no reason to want one. They put the genes in and switched them on. The recipient built magnetosomes, membrane compartments with magnetite crystals inside them, strung into chains along the axis of the cell. The cells became magnetically responsive. It wasn't a perfect copy of the donor. The crystals were smaller, and there were fewer of them per cell, and the researchers had to do real work on expression levels and iron supply to get it to run at all. Later work pushed the yields up considerably in an engineered version of that strain, but the fundamental result stands, and it's the closing argument. Take the genes out of context, put them in a stranger, and the stranger builds the organelle. That is what a solved system looks like. You know the parts, you know what each part does when you remove it, and you can reconstitute the whole thing somewhere it has never existed. There's a footnote to this that I like, because it complicates the story in a useful direction. In 2022, a team reported finding magnetosome gene clusters sitting in the genome of a phototrophic bacterium that doesn't make magnetosomes. The genes are there. The organism isn't using them. They're silent. And when the researchers manipulated the conditions, they could get that organism to start producing magnetosome-like particles. So possession of the cartridge and operation of the cartridge are separate questions. A genome can carry the instructions and never run them. That's a caution worth holding on to for later, when we start asking whether animals have anything like these genes. Because finding a sequence in a genome is a long way from finding a working machine in a cell. One more thing about the island before we go inside a single crystal. Magnetotactic bacteria don't form a taxonomic group at all. The label describes a behavior, and that behavior is distributed across a wide swath of the bacterial tree. You find magnetosome-forming organisms in the alpha proteobacteria, in the delta proteobacteria, in Nitrospirata, in candidate phyla that mostly exist as genome sequences from environmental samples because nobody has managed to culture them. Some of them make magnetite, some make greigite, an iron sulfide, which is chemically a completely different mineral produced under completely different conditions and which happens to also be magnetic. A few species make both in the same cell, in separate chains. The distribution of these genes across the tree and the pattern of their relationships has been read by several groups as evidence that magnetosome formation is old, possibly Archean, meaning billions of years old, meaning it may be one of the oldest forms of controlled biomineralization on this planet. Older than shells, older than bone, older than teeth. That interpretation is contested in the details, as everything about deep microbial phylogeny is contested in the details, so I'll flag it as a well-supported hypothesis and leave it there. But the broad point is robust. This is not a recent invention, and it has been moving around the bacterial world for a very long time. Which makes the animal situation stranger when we get there, because a trick that ancient, that transferable, that cheap in genes, and that useful is exactly the kind of thing you'd expect to find copied everywhere. Now, the crystal, a magnetotactic bacterium, is, by the standards of most living cells, absurdly full of iron. Depending on the species and the growth conditions, magnetite can account for somewhere in the low single digits as a percentage of the cell's dry weight. And estimates of intracellular iron content in these organisms run well above what a normal bacterium of the same size carries. They are hoovering iron out of their environment on a scale that would be toxic to almost anything else. And they're doing it in sediments where dissolved iron is frequently in short supply and chemically awkward, sometimes oxidized and insoluble, sometimes reduced and reactive. How exactly they get it in through the outer envelope is one of the parts of this system that's still murky, and it's worth saying so plainly. Different species seem to use different routes. Some evidence points to iron-scavenging molecules called siderophores. Some points to direct uptake of reduced ferrous iron. Some of the transport is probably shared with the ordinary iron metabolism every bacterium has. There isn't a single clean answer yet. Where it gets clean again is at the compartment wall. Getting iron from the cytoplasm into the magnetosome is the job of those Cation diffusion facilitator proteins MAM-B and MAM-M. Knock out MAM-M, and the cell still makes magnetosome compartments, and those compartments come out empty, or nearly so. The bag exists. The delivery system is broken. That's about as direct a demonstration of a transporter's function as you get in cell biology. And it also confirms the sequence of operations: compartment first, cargo second. Now the chemistry, which is where this gets specific in a way I think is underappreciated. Magnetite contains iron in two oxidation states at once. 2/3 of the iron atoms are in the ferric state, 1/3 in the ferrous state, locked into a crystal lattice in a fixed arrangement. If the cell simply pulled in ferrous iron and let it sit, oxygen would oxidize all of it, and you'd end up with one of the fully ferric iron oxides. Useless? If the cell kept everything reduced, you'd never form the mineral at all. So the cell has to hold a mixed oxidation state on purpose. It has to oxidize some of the iron and leave the rest alone inside a sealed compartment at a controlled pH continuously while the crystal grows. The proteins that appear to do this are one of the more elegant discoveries in the whole field. They belong to a family found, as far as anyone can tell, only in magnetotactic bacteria. Researchers named them magnetochromes. They're small domains built on the classic cytochrome plan, the same basic architecture that living things use everywhere to shuttle electrons, with an iron-containing heme group held in place by cysteine residues. 4 of the magnetosome proteins carry these domains: MAM-E, MAM-P, MAM-T, and MAM-X. Structural work published in Nature by a French group led by Marina Siponen, working with Pascal Arnoux and colleagues, cracked open what MAM-P looks like and what it does. The protein assembles into a shape they described as a nanoscale cage, with the magnetochrome domains positioned at the opening. In a test tube, they mise en marque they distes combatintu with no cell involved, MAM-P will take ferrous iron and oxidize it, producing exactly the kind of ferric mineral you'd need as a starting material. Mix it with ferrous iron under the right conditions and you get magnetite forming. So the compartment contains a dedicated electron handling system, whose job is to set the iron ratio. This is the redox thermostat. It's the reason the product is magnetite instead of rust. What happens next is the part that's still argued about. And the argument is worth understanding because it tells you how hard this is to study. The obvious model is that dissolved iron in the compartment simply reaches saturation, and magnetite precipitates directly out of solution. Simple, one step. The evidence says it's messier than that. Work led by Jens Baumgartner and Damien Favre's group, published in the Proceedings of the National Academy of Sciences, followed the mineral as it formed and reported that the cell first accumulates a phosphate-rich ferric hydroxide, a disordered gel-like iron phase with no real crystal structure, and that magnetite emerges out of that through intermediate iron oxyhydroxide stages. Other groups, using different methods on different species, have argued for ferrihydrite as the intermediate, or for something closer to direct precipitation. I don't think this is a case where one side is simply wrong. Different species, different growth conditions, different time resolution on the measurement. What everyone agrees on is that the cell maintains a reservoir of iron in a non-crystalline form and converts it, and that the conversion happens fast. When you supply iron to starved cells and watch with time-resolved techniques, magnetite signal appears within minutes. Then there's the question of where the crystal starts. A crystal has to nucleate. In a pure solution, nucleation is a probabilistic event. You need enough atoms to spontaneously arrange themselves into a stable seed, and that's a high-energy barrier, which is why supersaturated solutions can sit around doing nothing until something disturbs them. Biology gets around this the same way industrial chemistry does, with a template. A surface with the right geometry and the right charge distribution lowers the barrier and tells the crystal where to begin. That's the strongest candidate role for MMS6. It's a small protein, it self-assembles into groups, and it has a run of acidic amino acids at one end that binds iron ions. It's found bound tightly to the crystal surface. And in the test tube experiments, adding it to an iron solution changes the outcome. You get particles in a different size range and with different faces exposed. A protein that controls mineral formation with no cell around it is doing something structural and specific. MAM-E is also in this picture, and it's a peculiar protein because it carries a protease domain, meaning it can cut other proteins. Mutating that catalytic site produces cells that make small, immature crystals. So one of the controls on crystal growth appears to run through the cell, selectively chopping up its own magnetosome proteins. Regulation by demolition. Now step back and look at what comes out of the factory, because this is where the specification gets ruthless. Magnetosome crystals are chemically pure. Inorganic magnetite formed in sediments or in a lab beaker is full of substitutions, other metals sitting in the lattice where iron should be, plus defects, dislocations, twin boundaries. Biological magnetosome magnetite is close to stoichiometrically perfect and crystallographically near flawless. Rows and rows of atoms with essentially nothing out of place. They also have species-specific shapes. Magnetospirillum makes cuboctahedra, roughly isometric crystals with cube and octahedron faces. Magnetovibrio blakemorei, a marine species, makes elongated prisms. Several groups in a completely different bacterial lineage make crystals shaped like bullets or teeth, tapered at one end. Same mineral, different geometry. And the geometry breeds true. It's a species character, as reliable as any anatomical feature, which means it's under genetic control. That's harder than it sounds. Magnetite, left to its own devices, grows into an octahedron. That's the shape its lattice prefers, because that's the arrangement that minimizes surface energy. To get an elongated prism or a bullet, the cell has to override the crystal's own preference, suppressing growth on some faces, while permitting it on others. That means proteins binding specifically to particular crystal faces and blocking them. Face-selective inhibition at the scale of individual atomic planes. And here's the design choice that ties it together. When magnetosome crystals are elongated, the elongation runs along a particular internal axis of the crystal, and that axis is magnetite's easy axis of magnetization, the direction along which the magnetic moment naturally wants to lie. That isn't decorative. Stretching a magnetic particle along its easy axis increases what physicists call shape anisotropy. The energy cost of rotating the magnetization away from the long axis goes up, which means the crystal's magnetic direction becomes more strongly pinned. It resists being flipped by thermal energy. It stays a stable magnet at sizes where a round particle of the same volume would already be losing stability. The cell is elongating the crystal in the one direction that makes it a better permanent magnet. I want to be honest about how much of that is demonstrated and how much is inference. The crystallography is measured. The elongation direction is measured. The magnetic consequence of shape anisotropy is basic physics and not in dispute. Whether evolution shaped the crystal for that reason is an inference from function, and it's a strong one, because the alternative is a coincidence that keeps happening independently in unrelated lineages. So the shape is controlled, the purity is controlled, the mineral phase is controlled, that leaves size, and size is where this whole story stops being chemistry and becomes an engineering problem with a hard tolerance on both sides. Here's a fact about magnets that almost nobody is taught, and that turns out to govern this entire subject. A big piece of magnetic material is a bad magnet. Take a chunk of magnetite the size of a pea. Inside it, the atomic magnetic moments don't all point the same way. The material spontaneously breaks itself up into regions, called domains, and neighboring domains point in opposing directions. Why would it do something so self-defeating? Because a uniformly magnetized object projects a field out into the space around it, and that external field carries energy, a lot of it. The material can lower its total energy by splitting into opposed domains, so the field loops around internally and mostly stays home. The boundaries between domains, the walls, cost energy to maintain, so there's a trade-off. In a large piece, the savings win easily. The consequence is that a pea-sized lump of magnetite straight out of the ground has a net magnetic moment far, far smaller than the sum of its parts. Most of it cancels itself out. Now shrink the particle. As it gets smaller, the amount of energy you save by having domains falls faster than the cost of the wall that separates them. At some point, the wall isn't worth it anymore. Below that size, the particle gives up and magnetizes uniformly. Every atomic moment inside it lines up. The whole grain becomes one domain. That's the strongest permanent magnet you can make out of a given amount of material. Per unit volume, nothing beats it. A single domain particle of magnetite is magnetically saturated all the time, permanently, with no external field required, and it holds its direction for geological time. Single-domain magnetite grains in ancient rock are how we know the continents moved, because they recorded the direction of Earth's field when they formed, and they have not let go of it in hundreds of millions of years. For magnetite, the upper edge of that single-domain behavior sits somewhere around 80 to 120 nanometers, and exactly where it depends on the shape of the grain. Round, equidimensional particles lose stability at the low end of that range. Elongated ones stay single-domain up to larger sizes, which is precisely the trick we just watched the bacterium use. Above that upper edge, magnetite doesn't cleanly break into flat domains at these scales. It does something stranger. The magnetization curls into a vortex, a little whirlpool of magnetic direction inside the grain, with the moment mostly canceling around the loop. The particle keeps getting bigger, and its usable magnetic moment barely grows. You spend iron, you spend energy, you spend membrane, and you get almost nothing in return. That's the ceiling. Now the floor, which is more brutal. Go the other way. Shrink the particle below the single-domain range, down toward 30 nanometers and under. The particle is still uniformly magnetized. Every atom still lines up. But the total energy holding that magnetization pointed in any particular direction scales with the volume of the grain, and volume falls off as the cube of the size. Halve the diameter, and you have 1/8 the volume, and roughly 1/8 the anisotropy energy, pinning the direction in place. At some point, that pinning energy drops to the same order as the thermal energy in the environment. And then the whole magnetic moment starts flipping, spontaneously, randomly. The direction reverses, then reverses again, driven by nothing but the ambient heat on a timescale that can drop to billionths of a second. Physicists call this superparamagnetism, and it's worth being precise about what it means, because this specific piece of physics is going to matter enormously later in this story. A superparamagnetic particle is not weakly magnetic. Instantaneously, it's strongly magnetic. What it lacks is memory. Averaged over any timescale a cell cares about, its direction is 0. It's a compass needle that spins 1 billion times a second. Put it in a magnetic field, and it will respond, statistically, with a slight bias toward alignment. Take the field away, and it retains nothing. Superparamagnetic particles are useful for some things. They can be pulled around by a strong field gradient. They show up all over medical imaging and laboratory separations. What they cannot do is tell you which way is north on their own, because they have no persistent direction of their own to compare against anything, so there's a window. Above roughly 30 nanometers, below roughly 120, with the exact edges depending on shape. Inside the window, a stable, permanent magnet. Below it, a magnet with amnesia. Above it, a magnet that cancels itself out. Every magnetosome crystal ever measured across every species sits inside that window. That statement sounds like an overreach, and it's close to being one. So let me put it carefully. When you measure thousands of magnetosome crystals and plot their sizes, you get a distribution with a sharp shape, tight, narrow, cut off abruptly at the large end, with a tail that runs down towards smaller sizes, which is what you'd expect from a population that includes some crystals that are still growing. The mean sits comfortably in the stable single-domain range for that species' crystal geometry. Compare that to magnetite formed by ordinary geological or chemical processes. That gives you a log-normal distribution, a long, ugly tail out to large sizes, a broad spread, no characteristic value. Inorganic crystal growth is a process where big crystals keep eating small ones, and the distribution smears out over orders of magnitude. The bacterial distribution looks like a manufactured part. The inorganic one looks like gravel. Let me put the tolerance in terms a machinist would recognize, because that's really what this is. A magnetite crystal 50 nanometers across is about 120 unit cells wide. Roughly 120 repeats of the basic atomic building block, end to end. The lower edge of usefulness sits at maybe 70 unit cells. The upper edge at maybe 250. The cell is controlling the dimension of a solid object to within a few dozen atomic layers in a wet compartment by protein chemistry without measuring anything in the dark at the bottom of a pond. What gets me is that this is done with a dial. Remember the 4 genes of that second operon, the ones that are partly interchangeable, where taking all 4 out shrinks the crystals to about 3/4 of normal, and putting any one of them back pushes the size partway home. That behaves less like a switch and more like a rheostat built out of protein abundance. The cell is regulating crystal size the way you'd regulate a thermostat, by adjusting how much of a certain something is present in the room. Now here's the arithmetic that makes the whole thing hang together, and I'll do it slowly because it's the load-bearing calculation in this entire field. Take one magnetosome crystal, 50 nanometers on a side. Magnetite has a known saturation magnetization, the magnetic moment packed into each unit of volume, and it's a standard measured value. Run that through the volume of the crystal, and you get its total magnetic moment. Then put that moment in Earth's magnetic field, and the alignment energy, the energy difference between pointing along the field and pointing across it, comes out at roughly 3 × 10 to the minus 21 joules. Now compare that to the thermal noise floor, about 4 × 10 to the minus 21 joules. The crystal loses. One magnetosome, all by itself, sitting in a cell in Earth's field, gets shoved around by thermal collisions harder than the field pulls on it. Its orientation would be mostly random. It would be an unreliable compass, giving a weak statistical bias at best. That single number explains the architecture of the entire organism. It's why there's a chain. It's why the chain has 15 or 20 or 30 crystals in it instead of 1. Because magnetic moments in a line, all pointing the same way, add. 20 crystals give you 20 times the moment, and 20 times 3 parts against 4 puts you at something like 14 or 15 times the thermal floor. That's the design margin, roughly an order of magnitude over noise. Enough that a cell swimming in open water holds its heading against thermal buffeting with an average alignment on the order of 90%, and not so much that it's wasting iron on overkill. There's a forensic consequence to all of this that's going to become the center of the second half of this story, so I want to plant it here properly. Because biological magnetite has such a distinctive signature, a narrow size distribution, a stable single-domain range, chemical purity, near-perfect crystal structure, unusual shapes, elongation along the easy axis, and arrangement in chains, Joseph Kirschvink at Caltech proposed that you could use those features as a test. A set of criteria that, taken together, would identify magnetite as biological in origin even with no cell left around it, no DNA, no membrane, nothing but the mineral. That idea had an obvious application. Magnetofossils, chains of bacterial magnetite that survive in sediment after the cell that made them is long gone, preserved as a record of ancient microbial life. It had a much less obvious application too, and this is one of those places where two fields collide in a way nobody planned. In the late 1990s and early 2000s, those same criteria were dragged into the middle of the argument over ALH 84001, the Martian meteorite recovered from Antarctica that a NASA team claimed contained evidence of past life. A significant part of that claim rested on magnetite crystals inside carbonate globules in the rock, and on the argument that some of them matched the biogenic profile, the right size range, the right shapes, elongated along the right axis, arranged in what looked like chains. That claim did not survive. The consensus today is that the Martian magnetite can be explained without invoking life, and the meteorite is no longer treated as evidence for anything except how hard this kind of inference is. But the criteria survived, and they're the reason that, when someone announces they've found magnetic material inside an animal's tissue, the immediate and correct response from anyone who knows this literature is a question, and it lands with a certain flatness, which is fine. What's the size distribution? So the cell now has 20 permanent magnets inside it. Small, pure, single domain, each one in its own membrane compartment. That is a problem, because permanent magnets attract each other, and at these distances they attract each other hard. Two magnetite crystals, sitting 15 or 20 nanometers apart in a watery compartment, exert forces on one another that are large compared to anything else acting at that scale. And the arrangement they want to reach, the one that minimizes their combined magnetic energy, is not a straight line. Magnetic particles left to themselves in a confined space collapse into compact clusters, into closed loops, into flux-closed rings where each particle's north end feeds into the next particle's south end and the whole assembly's field stays internal, which cancels the moment. The clump has almost no external magnetic direction. It's the same self-defeating behavior as the domains in the pea-sized lump, just built out of separate particles instead of separate regions. So a straight chain is unstable. It's an arrangement the physics actively fights, to hold 20 magnets in a line, end to end, all pointing the same way. Something has to be physically restraining them. That something turned out to be a cytoskeleton in an organism that was not supposed to have one. For most of the 20th century, the cytoskeleton was another of those things that separated us from bacteria. Eukaryotes have actin and tubulin, filaments and microtubules, an internal scaffolding that gives cells shape and moves things around inside them. Bacteria were held to be too small and too simple to need any of that. That view came apart in the 1990s and 2000s, and magnetotactic bacteria were part of what took it apart. There's a gene in the core operon called MAMK. Its sequence is recognizably related to a bacterial protein called MARB, which is itself a structural relative of actin. Actin, in your cells, polymerizes into filaments that form the mechanical framework of the cell. MAMK does something similar. It polymerizes into long filaments that run the length of the bacterium. When Comelle and Jensen looked at their cryotomograms, they could see them. Filamentous structures running alongside the magnetosome chain, straight, spanning much of the cell. Delete MAMK, and the filaments are gone, and the magnetosome chain falls apart. The compartments still form. The crystals still grow. The organization goes. Then there's MAMJ. And the MAMJ experiment is the one I'd put in a textbook if I were writing one, because it's a case where you can predict the result from physics before you run it. The MAMJ protein is highly acidic, with a long repetitive stretch in the middle, and it sits on the magnetosome membrane. The proposal was that it acts as a connector, physically linking each magnetosome vesicle to the MAMK filament, so the chain is clipped to the rail. Delete MAMJ. What should happen? If MAMJ is the clip, then cutting it releases 20 magnets that are still made, still magnetic, still in the cell, and now free to move. And free magnets collapse. Work from Schuler's group published in Nature reported exactly that. In cells lacking MAMJ, magnetosomes formed normally and then aggregated into compact clusters. The chain was gone. In its place, a huddle of magnetite crystals doing precisely what a physicist would tell you a set of unrestrained nanomagnets in a confined volume will do. I find that result more convincing than 100 correlations. The prediction came from the physics, the experiment matched it, and the mechanism it implies is mechanical, a tether, holding an unstable arrangement in place against a force that wants to destroy it. And this is where the design starts to feel deliberate in a way that's a little uncomfortable, because there's another reason the tether matters, and it has nothing to do with keeping the chain straight. Think about what the cell needs to happen when it encounters a magnetic field. It needs to turn. The whole cell, body, flagella, everything, so that when the flagella push, they push along the field line. Now suppose the magnetosome chain were floating loose in the cytoplasm, still a straight chain, just unattached. Put that cell in a magnetic field. The chain would rotate to align with the field. The cell around it would not. The chain would sit there, aligned, inside a body, pointing whatever direction it was already pointing, and the organism would swim off in a random direction with a beautifully oriented compass rattling around inside it, doing nothing. Torque has to be transmitted. The magnetic field applies a twisting force to the chain, and that twisting force is only useful if it's coupled to the cell wall. The filament is the coupling. MAM-K anchors to the cell envelope. MAM-J clips the magnetosomes to MAM-K, and the result is a rigid mechanical linkage from crystal to cell body. The whole bacterium becomes the needle. There's a further refinement that I love for how fussy it is. Many of these organisms are spirilla. They're curved, helical cells, shaped like a corkscrew. If you laid a chain of magnets down inside a corkscrew and simply followed the wall, the chain would be curved too. And a curved chain of magnets has a weaker net moment along the swimming axis than a straight one. The cells don't do that. The chain runs straight along the shortest path through the curved body, what geometers would call the geodetic axis. Work from Schuller's lab identified the protein MAM-Y as involved in this, positioning and constraining the chain so that it takes the straight route through a body that isn't straight. The cell is building a rigid straight line inside a spiral. Somebody, a very long way back down that lineage, optimized for the last few percent. Then there's reproduction, which is a properly awkward engineering problem that I don't think most descriptions of this system take seriously enough. The chain sits near the middle of the cell. When the cell divides, it divides in the middle, straight through the chain. So every division, this organism has to cut its own compass in half and give a working piece to each daughter. And the daughters then have to reposition the fragment they inherited back to the center of their new, smaller selves and extend it, and keep the magnetization of the new crystals aligned with the old ones. Studies of dividing cells show the chain being split and the halves being actively moved, with the cytoskeletal machinery involved in the repositioning. And there is a quietly clever bit of housekeeping in all of this. When a new magnetosome starts growing at the end of an existing chain, it's growing inside the magnetic field produced by the crystals already there. That field is strong at that distance, so the new crystal, as it passes through the size, where it becomes magnetically stable, gets locked in pointing along the chain, because that's the field it grew up in. The chain magnetizes its own new members. Nobody has to check. Nobody has to correct anything. The geometry does the alignment for free, which is exactly the kind of solution evolution finds. And engineers usually don't, because engineers tend to add a sensor where physics would have done the job. Put the whole assembly together, and you can measure what it produces. The magnetic moment of a single magnetotactic bacterium comes out somewhere in the region of 10 to the minus 15 ampere meters squared, varying with species and crystal count. That is a vanishingly small magnet by any human standard. It's also, as we worked out, roughly 10 to 15 times the thermal noise floor when placed in Earth's field. And that ratio is the only number that matters. Drop one of these cells into a beaker with a field applied, and it aligns in a fraction of a second. Reverse the field, and it turns around physically as a body and swims the other way. That's the behavior Blakemore saw on his slide, and it comes out of a stack of design decisions that runs from the redox chemistry inside a nanoscale membrane sac, all the way up to a cytoskeletal filament pinned to the cell wall. Membrane compartment, iron transporter, redox control, templated nucleation. Size held inside a physical window with a tolerance of a few dozen atomic layers. Shape elongated along the easy axis. 20 of them, in a chain, tethered to a filament, running the straight path through a curved body, magnetized in series, split and rebuilt, every generation. That's the specification. That's what a biological compass costs. And now I have to tell you the thing that makes the whole picture turn sideways, because for all of that machinery, for all 30 genes and the organelle and the cytoskeleton, these bacteria cannot navigate, not even a little. Here is the experiment that settles it, and it's about as low-tech as science gets. Take a culture of magnetotactic bacteria, kill them, fix them in alcohol or formalin, the way you'd preserve a specimen, so that every enzyme is denatured, every membrane potential is gone, every metabolic process has stopped. There is no biology happening in those cells anymore. They are objects. Put them under the microscope with a magnet. They align. Every dead cell in that preparation swings around and points along the field, exactly as it did when it was alive. It doesn't swim, obviously. It just sits there, oriented, like iron filings on a sheet of paper over a bar magnet, because functionally, that is what it now is. That single observation tells you what the magnetosome chain is and what it isn't. It isn't a sense organ. Nothing about the alignment requires the cell to detect anything, process anything, or decide anything. The chain is mechanically coupled to the cell body. The field applies a torque to the chain. The body turns. That's the entire mechanism. It would work in a dead cell, in a plastic model, in a grain of sand with a magnet glued inside it. The word magnetotaxis is therefore a little misleading, and the people who work on this know it and use it anyway because the name stuck. Taxis, in biology, usually means a directed response to a stimulus. A bacterium doing chemotaxis is actually sampling its environment, comparing concentrations over time, and changing its swimming behavior based on the comparison. There's a sensory system, a signaling cascade, a decision. Magnetotaxis has none of that. The cell is a compass needle with a motor on the back. So what is it for? These organisms live in a very specific and very thin place. Go out to a coastal pond or a salt marsh or a stratified basin and take a core down through the water and into the sediment. Oxygen concentration falls as you go down. Near the top, the water is oxygenated. Below a certain depth, oxygen runs out entirely, and the chemistry turns reducing, full of sulfide and reduced iron. In between, there's a boundary layer where oxygen is present but scarce. In some settings, that layer is centimeters thick. In others, it's millimeters. Magnetotactic bacteria are microaerophiles. They want oxygen, but only a little of it. Too much, and it's toxic to them. Too little, and they can't make a living. Their preferred habitat is that thin band, and their whole existence is about finding it and staying in it. Now think about the search problem from inside a cell that's 2 microns long. You're trying to find a horizontal layer of specific chemistry in a 3-dimensional volume. You have a flagellum, so you can swim. You have chemical sensors, so you can tell whether conditions are getting better or worse, but you have no idea which way is up. That's the thing people forget. You're too small to feel gravity in any useful way. The density difference between you and the water around you is tiny. The buoyant force is tiny. And Brownian motion is throwing you around constantly. Gravity, at your scale, is noise. So a random 3-dimensional search it is. Swim, sample, turn, swim, sample. In 3 dimensions, with random turns, that's a slow and inefficient way to find a layer, unless you can find a vertical reference. And Earth's magnetic field, almost everywhere on the planet, has one. The field doesn't run flat along the surface. It comes out of the ground in the southern hemisphere, arcs over, and dives back into the ground in the northern hemisphere, and the angle it makes with horizontal is called the inclination. At the magnetic equator, the field is horizontal, and the inclination is 0. Over most of the United States, the field dips downward at something like 60 to 70 degrees. Near the poles, it's close to vertical. So over most of the planet, aligning with the field line means aligning with something that has a strong vertical component. Follow it one way, and you're heading down. Follow it the other way, and you're heading up. That converts a three-dimensional search into a one-dimensional one, which is an enormous simplification. The cell picks a direction along its axis, swims, samples oxygen chemically, and if the oxygen is wrong, reverses, up, down, along one line, until it finds the band it wants and settles in. Richard Frankel and Dennis Basilinsky and colleagues formalized this as magnetoaerotaxis, and they identified more than one behavioral variant. Some species swim persistently in one direction and reverse only when the chemistry demands it, while others run back and forth along the axis in both directions like an ordinary chemotactic bacterium that happens to be constrained to a rail. What the magnetic field supplies here, in other words, is verticality. It stands in for a sense of up in an organism too small to have one. And there's a test of that idea, and it's the kind of test that makes you like science. If the field is being used as a vertical reference, then the polarity has to be different in different hemispheres. In the northern hemisphere, the field dips down toward the north, so a cell that swims toward magnetic north is swimming down. In the southern hemisphere, the field dips down toward the south, so a cell that wants to go down has to swim toward magnetic south. Same behavior, opposite polarity, and the switch has to happen at the equator. Blakemore, Frankel, and Ralph Wolf went and checked. They collected in the southern hemisphere, in New Zealand, and reported that the populations there were predominantly south-seeking, the mirror image of what they'd been finding in Massachusetts. Later sampling near the geomagnetic equator turned up roughly comparable numbers of both polarities in the same water, which is what you'd predict in a place where the field is horizontal and neither direction takes you down. That's a prediction, a field expedition, and a confirmation. It's clean. It's the kind of result that makes a hypothesis feel earned. I do want to complicate it, though, because the current literature has gotten less comfortable with this story than the textbook version suggests, and a version that only reports the tidy parts isn't worth much. The magnetoaerotaxis model works beautifully for cells swimming freely in open water. Most magnetotactic bacteria, most of the time, live in sediment, and sediment is a maze. It's packed with mineral grains, it's viscous, it's tortuous, and a cell in there is not swimming down a clean field line. Measurements of the actual mean magnetic alignment of these bacteria within sediment have come out startlingly low, in one study under 1%, which would strip away most of the theoretical navigational advantage the model depends on. On top of that, researchers keep finding cells of opposite polarity coexisting at depths that don't line up with the redox gradient the way the model predicts. So the mechanism is solved, and the ecology is still argued about. Those are different questions, and it's worth keeping them apart. We know exactly how the cell builds the magnet and exactly how the magnet turns the cell. What that buys the organism in its real habitat is a live research question with real disagreement in it. And that sets up the hinge of this whole subject, which is a thing I don't think gets said often enough or plainly enough. The bacterium never solved the hard problem. Everything we've walked through, the compartment, the gene island, the redox thermostat, the templated nucleation, the size window, the chain, the tether, all of it delivers exactly one output, a body that points along a field line. There is no signal. Nothing is measured. No information about the magnetic field ever enters the cell as information. The field acts on the cell the way wind acts on a weather vane, and the cell's chemical senses do the rest of the work. An animal cannot use that. A loggerhead turtle does not need its body physically rotated by the geomagnetic field. Its body is too large for a nanoscale magnet to move by an almost unimaginable margin, and even if you could scale the magnet up, being passively swung around by the field is useless to an animal that needs to make decisions. A turtle needs to know the field's direction and, for the map sense, its intensity and inclination, as neural information, numbers in a nervous system, something it can compare, remember, and act on days later. That requires a transducer, a structure that converts magnetic torque or magnetic force into a mechanical deformation, that deformation into a change in an ion channel, that change into a shift in membrane voltage, and that voltage into a train of nerve impulses going somewhere in the brain. None of that exists in the bacterium. It never had to. Its entire body was small enough to serve as the moving part. So the solved case gives us a manufacturing specification and stops one step short of the thing animals actually need. It tells us how to build the magnet. It says nothing about how to wire it. And that's the gap the search has been trying to close for 50 years. Let's write the specification down because from here on, everything gets measured against it. And the discipline of having it written down is most of what separates a finding from an announcement. For a magnetite-based magnetic receptor to exist in an animal, 5 things have to be true at the same time. 1. There has to be magnetic material inside a cell. Inside. Not on it, not near it, not in the tissue around it. And the material has to be in the stable single-domain range, or a chain of superparamagnetic particles arranged so tightly that they behave collectively as one, which is a scheme people have proposed and which has its own physics. The material has to be biogenic, meaning the animal made it, which, given everything we've said about crystal habits and size distributions, is something you can actually test. 2. The magnetic material has to be mechanically coupled to something. A permanent magnet in a cell, floating free in the cytoplasm, does nothing. It has to be tethered to a structure that can transmit force, a cytoskeletal element, a membrane, a filament attached to an ion channel. The whole game is converting a torque into a strain. 3. The cell has to be a sensory cell. It needs to be innervated, or it needs to be a neuron itself, or it needs a synaptic output onto a neuron. A cell full of magnetite with no way to tell anybody about it is a storage depot. 4. There has to be a nerve carrying a signal that changes when the magnetic field changes, and that signal has to be recordable, so somebody can put an electrode on it, sweep the field, and watch the firing rate move. 5. Take the structure away, and the behavior has to break. Put it back, or spare it. And the behavior has to work. That's the loss of function test. And in the bacterium, it's trivial. Delete MAM-J, the chain collapses, the cells stop responding to the magnet. Done in an afternoon. 5 requirements. In magnetotactic bacteria, requirements 1 and 2 are satisfied in overwhelming detail. 3 and 4 don't apply because the organism doesn't use nerves. And 5 has been demonstrated dozens of times over. In animals, we have never satisfied all 5 for any candidate structure. Not once. In half a century. Before we go into why, I want to deal briefly with the obvious objection, which is that maybe animals don't use magnetite at all. That's a fair objection, and there are 2 other routes on the table, both of them real. The first is chemical. There's a class of light-sensitive reactions where absorbing a photon creates a pair of molecules, each carrying an unpaired electron. And for a brief window, the spins of those 2 electrons are quantum mechanically correlated. The rate at which they interconvert between different spin states is sensitive to magnetic fields, including fields as weak as Earth's. And the yield of the reaction products therefore depends on the angle between the molecule and the field. Put enough of those molecules in an ordered array in a retina, and you have, in principle, a chemical compass that paints direction onto vision. The candidate molecule is a protein called cryptochrome, found in the eyes of migratory birds, and there's a substantial and increasingly sophisticated body of work behind it. That mechanism has a signature. It gives you an axis without a polarity, meaning it can tell you where the field line runs without telling you which end is north. It requires light, and it can be disrupted by extremely weak oscillating radio frequency fields, which interfere with the spin dynamics and which have essentially no effect on a lump of magnetite. That last property is the sharpest tool anyone has in this field for telling the two mechanisms apart, and we'll use it later. The second route is induction. Move a conductor through a magnetic field, and you generate a voltage. Sharks, skates, and rays swim through seawater, which conducts, and they carry the most sensitive electroreceptors in the animal kingdom, the ampullae of Lorenzini, gel-filled canals in the head that detect field gradients down to a few nanovolts per centimeter. Adrianus Calmine proposed decades ago that these animals could, in principle, derive magnetic direction from the voltages induced by their own motion. It's physically sound. Whether they actually do it is still unsettled. So this is a subject with several live possibilities. And I'm not going to pretend the magnetite route is the only game or that it's one. What I'll say is this: the magnetite route is the only one that requires an object. Chemical magnetoreception is a property of molecules that are already there doing other things, distributed through tissue, invisible to any anatomical search. Induction needs no dedicated hardware at all, beyond electroreceptors that already exist for other purposes. Magnetite is the only hypothesis that predicts a thing, a manufactured, mineral, physically locatable thing, with a size distribution and a crystal habit and a place in a cell, which means it's the only one you can go looking for. And that is exactly why the search for it has generated so much drama. Now, the practical problem: how many cells would a magnetic receptor need? Probably very few. Run the physics, and you find that a modest number of well-coupled receptor cells, arranged with known orientations relative to the body, would be enough to extract direction and, potentially, intensity. Estimates that get thrown around sit in the hundreds to low thousands. Some proposals get by with fewer. A pigeon has many billions of cells. A loggerhead turtle has vastly more. You are looking for something on the scale of 1,000 cells, of unknown appearance, somewhere in a body, and you don't have an antibody that stains them. You don't have a gene that marks them, and you don't have a shape to look for. What you have is iron. That's the whole toolkit for the anatomical approach. You stain for iron, you look for cells that light up, and you follow the ones that look interesting. And iron in an animal body is everywhere. Hemoglobin is iron. Myoglobin is iron. Ferritin, the universal iron storage protein, is present in essentially every cell type in the body and forms iron oxide cores inside its protein shell. Macrophages that recycle worn-out red blood cells are absolutely loaded with iron, sometimes visibly brown with it under the microscope. Blood in a capillary is iron. A speck of dust from a steel dissecting tool is iron. Staining for iron in animal tissue and looking for a magnetoreceptor is like trying to find a specific grain of sand by searching a beach for things made of silicon. Then there's the second problem, and this one is structural, and it is not going away. You cannot do genetics on a sea turtle. In the bacterium, the entire mechanism was cracked open by knocking out genes one at a time and looking at what broke. That approach requires an organism you can culture in enormous numbers, transform with foreign DNA, and screen rapidly, and where the phenotype you're screening for shows up under a microscope in an afternoon. Loggerheads take decades to mature, are protected under law nearly everywhere they occur, and their magnetic behavior is measured by watching hatchlings orient in a tank of water surrounded by coils. The standard genetic model organisms don't rescue you either, because their magnetic behavior is either absent, weak, or contested. There's a long and unhappy literature on magnetic responses in fruit flies and in laboratory rodents, with results that come and go between labs. The one place where behavior is rock solid, in the long-distance migrants and the ocean navigators, is precisely where the genetic tools are worst. So the field is stuck with two approaches, and both are indirect. Approach 1. Follow the iron. Take the tissue, stain it, section it. Look for magnetic material, characterize it, and try to work out whether the thing you found is a receptor. This is the anatomical route, and it has produced the two most famous failures in the subject. Approach 2. Follow the nerve. Skip the cell entirely. Put an electrode on a nerve, change the magnetic field, and see whether the firing rate changes. Or go coarser, cut a nerve, or anesthetize it, or ablate a brain region, and see whether the animal's magnetic behavior breaks. This route can't identify the receptor. What it gives you is a rough location and a list of connections, which is progress of a kind. There's a third approach that's really a physics trick, and it's the sharpest thing anyone has: the magnetic pulse. Hit an animal with a brief, intense magnetic pulse, oriented across the direction of its normal magnetization, strong enough to remagnetize single-domain particles. If the animal has permanent magnetic material in a receptor, the pulse should scramble what that receptor is reporting. If the animal is using a chemical compass or induction, a brief pulse should do nothing at all. Then release the animal and see what it does. That experiment has been run on birds, on bats, on sea turtles, on spiny lobsters, on salmon. We'll get to what it found. But the anatomical route came first, and it's where the case files start. The first one opens in a pigeon's face. The idea that animals might be carrying magnetite got serious in the late 1970s, and it got serious fast. In 1978, a team including James Gould, Joseph Kirschvink, and Kenneth Defies reported magnetic material in honeybees. The following year, magnetite turned up in the heads of homing pigeons, then in tuna, in the ethmoid region of the skull, then in salmon, then in cetaceans. Magnetometry on tissue samples kept returning permanent magnetic moments where there shouldn't have been any, and the samples were coming from exactly the animals that behavioral biologists already suspected of using the geomagnetic field. In 1981, Kirschvink and Gould published a theoretical paper working out how a magnetite-based receptor could function, what size the crystals would need to be, how they could be coupled to a mechanically gated ion channel, and whether the energies involved could clear the thermal noise floor. Their conclusion was that the numbers worked. A properly built magnetite receptor was physically allowed. So by the early 80s, you had a plausible mechanism, a physical theory, and magnetic material showing up in tissue from multiple species. The obvious next step was to find the cell. And for a while it looked like somebody had. Beginning in the early 2000s, Gerda Fleissner and Guenther Fleissner in Frankfurt, working with colleagues, published a series of papers describing iron-rich structures in the upper beak of homing pigeons. The descriptions were detailed and specific in a way that made them extremely persuasive. The structures sat inside the dendrites of the ophthalmic branch of the trigeminal nerve, which is the big sensory nerve of the face. There were said to be 6 discrete regions arranged bilaterally, in a consistent 3-dimensional pattern. The iron was reported in 2 forms: platelets of magamite, which is a magnetic iron oxide closely related to magnetite, and clusters of superparamagnetic particles. The proposal was that these arrangements were oriented in 3 perpendicular directions, allowing the bird to resolve the field as a vector. There was nothing vague about it. It had locations, it had structures, it had a mechanism, it had a nerve attached to it. It was an anatomy. It also had support from an entirely different direction. In 2004, Cordula Mora and colleagues published work in Nature in which pigeons were trained to discriminate the presence of a strong magnetic anomaly in a testing chamber. The birds learned the task, then the researchers interfered with the trigeminal system, cutting the ophthalmic branch or applying a local anesthetic to the upper beak, and the birds could no longer do it. Attaching magnets to the upper beak also broke performance, while attaching brass weights of the same mass did not. So there was a structure in the beak, a nerve leading from it, and a behavioral experiment showing that disabling that nerve destroyed a magnetic discrimination. That combination went into textbooks, it went into review articles, it went into every popular account of animal navigation written between roughly 2005 and 2012. If you read anything about how birds sense magnetism in that window, you read about iron in the beak. Then a group in Vienna decided to do the boring version of the experiment. David Keyes and his lab, with Christoph Treiber as first author, set out to map the iron-rich cells in the pigeon beak properly and systematically. Not in a few birds, in many. And with the full toolkit, magnetic resonance imaging and computed tomography to build a three-dimensional reference of the beak, then serial sectioning and staining, then electron microscopy of the cells themselves, then immunohistology to ask what kind of cells they actually were. The results, published in Nature in 2012, dismantled the whole thing. First, the distribution. If these cells are sensory receptors, they should be in consistent places, in consistent numbers, bird to bird, the way rod cells are in consistent places in a retina. They weren't. The number of iron-rich cells varied between individual pigeons by orders of magnitude. Their positions varied. Some of them were in the beak tissue. Some of them were somewhere else entirely. There was no reliable pattern, no bilateral symmetry of the kind that had been described, no 6 regions. That alone should stop you. Sensory systems are stereotyped. Development builds them to a plan. A receptor organ whose cell count swings by a factor of 100 between healthy individuals of the same species is not doing sensory work. Second, the ultrastructure. When they looked inside these cells with electron microscopy, they found ferritin-like granules. They found siderosomes. They found hemosiderin, which is what you get when iron storage overflows and gets dumped into lysosomes. And they found filopodia, the crawling extensions that motile cells use to move through tissue. That is a portrait of a macrophage. Specifically, an iron-loaded macrophage, the kind that shows up wherever tissue is breaking down red blood cells or clearing damage. Third, the conformation. They ran immunohistology against major histocompatibility complex class II, which is a marker displayed by antigen-presenting cells of the immune system. The iron-rich cells lit up. They were immune cells. They had always been immune cells. Every one of them. The beak of a pigeon is an organ that spends its life pecking gravel, and pigeons in general carry a lot of iron. A tissue that gets minor mechanical damage constantly and that has iron in circulation is going to accumulate exactly this population of cells, in numbers that vary with the individual bird's history, in locations that vary with where it happened to get knocked about. The Prussian blue stain, which is the classic histological method for showing iron, and which had been used throughout the earlier work, does not distinguish an iron-rich macrophage from a hypothetical iron-based receptor. It just shows iron, and iron-rich macrophages are far, far more common in tissue than magnetoreceptors could possibly be. The Fleissners disputed the reinterpretation, and there was an exchange in the literature about it, as there should be. But the field moved. The beak receptor came out of the textbooks. I want to be precise about what this did and didn't kill because this is exactly the place where popular accounts overshoot. It killed a cellular candidate. Trigeminal magnetoreception survived it intact. The behavioral and neurophysiological evidence that the trigeminal nerve carries magnetic information in birds has actually held up rather well and has gotten better. Work on Eurasian reed warblers from Dmitry Kishkinev, Nikita Chernetsov, Henrik Moritzen, and colleagues showed that when you deactivate the ophthalmic branch of the trigeminal nerve, the birds lose the ability to correct for a virtual magnetic displacement. Take a migrating warbler, put it in a field that simulates being about 600 miles east of where it actually is, and a normal bird will compensate and shift its heading. A bird with the trigeminal branch disabled will keep heading the way it was already going. It still has its compass. What it's lost is its position. And in a separate and beautifully designed experiment led by Manuela Zapka in Moritson's group, published in Nature, European robins with the trigeminal ophthalmic branch cut could still orient with a magnetic compass. Their compass was fine. Cutting a different pathway, the one running from the eyes to a forebrain region called cluster N, broke the compass instead. So the picture that emerged is a split system. Two magnetic senses, doing different jobs, running through different anatomy, a compass associated with the visual system, and a map or positional sense associated with the trigeminal nerve. And that trigeminal input remains the most likely place in a bird for a magnetite-based detector to live. The receptor at the end of that nerve has still never been found, which is a strange and slightly maddening position for a field to be in. You know the wire. You've cut the wire and watched the function disappear. You've traced the wire toward the front of the animal's head. And when you get to the end of it and look at what's there, all you can find is immune cells doing housekeeping. That was case file number 1. The second one is in a fish, and it's worse. Because in the second one, the contamination was in the laboratory. In 1997, a paper appeared in Nature that looked, at the time, like the answer. Michael Walker, Carol Diebel, Joseph Kirschvink and colleagues had been working on rainbow trout, and what they published was the most complete package anyone had ever assembled for a vertebrate magnetic sense. It had 3 legs, and each leg was independently strong. The first leg was behavior. They conditioned trout to respond to a change in magnetic field intensity, using food as the reward. The fish learned it. That establishes, without ambiguity, that the animal can perceive the stimulus. The second leg was electrophysiology. They recorded from a specific nerve, the superficial ophthalmic branch of the trigeminal, which in fish anatomy gets abbreviated as ROS5, and they found single units in that nerve whose firing changed when the magnetic field changed. That's a signal on a wire, which is requirement number 4 on our list. The third leg was anatomy. They traced the responsive units back toward their origin, and the trail led to the olfactory lamellae, the folded sheets of tissue in the nose. There, in a discrete sublayer, they identified candidate cells containing iron-rich crystalline inclusions, behavior, physiology, anatomy, in one animal, in one paper. 3 years later, in 2000, Diebel, Kirschvink, Walker, and colleagues followed it up with a second Nature paper that pushed harder on the material itself. They used magnetic force microscopy, which images magnetic fields at the nanoscale, together with electron microscopy, and reported that the inclusions in those candidate cells were magnetite, that the crystals were in the single-domain size range, and that they were arranged in chains. The title of that paper was as confident as titles get. Magnetite defines a vertebrate magnetoreceptor. If you had asked me in 2001, on the evidence available, I'd have said the trout case was closed, and the rest was cleanup. The trouble with the anatomical approach, as the pigeon story showed, is that you find what you stain for. So a group of researchers came up with a way around it. And it's a good idea, and I want to give it credit before I tell you where it went. The idea is stop staining. Screen by physics. If a cell contains a permanent magnet that's mechanically coupled to its structure, then that cell, floating free in a dish, should rotate when you rotate a magnetic field around it. Nothing else in a cell suspension should do that. You don't need to know what the receptor looks like. You don't need an antibody. You just need a rotating field and a microscope, and you let the physics pick the cells out for you. In 2012, Stefan Eder, Hervé Cadieux, Michael Winkelhofer, Joseph Kirschvink, Peter McNaughton, and colleagues published exactly this in the Proceedings of the National Academy of Sciences. They dissociated trout olfactory epithelium into single cells, put the suspension in a slowly rotating magnetic field, and watched. A small number of cells rotated. Rare ones, on the order of 1 in tens of thousands, which is roughly the abundance you'd expect for a specialized receptor. Those cells tracked the field as it turned. From the rotation dynamics, the team could estimate the magnetic moment of each cell, and the values came out large. Considerably larger than a bacterium-style chain of 20 crystals would give you. The magnetic material also appeared to be firmly coupled to the cell, at or near the membrane, which is what a functional receptor would require. It was a clever method, and it produced candidate cells that behaved physically the way a magnetoreceptor should behave. Then came 2015. A team including Nathaniel Edelman and Stefan Eder, working with David Keyes and with Kirschvink and Winkelhofer among the authors, took the next step. They ran the same rotating field screen, isolated the cells that responded, and then did something the earlier work hadn't. They took those individual identified cells and put them through correlative light and electron microscopy. You mark the exact cell you saw rotating, you fix it, you section it, and you look inside that specific cell with an electron beam. If the magnetic material is a biogenic intracellular receptor structure, it's in there. It has to be. That's the whole hypothesis. They couldn't find it. The paper, published in PNAS, reported no evidence of intracellular structures consistent with biogenically derived magnetite in the cells the screen had selected. The cells had large magnetic moments. Something magnetic was there. What they could not find was material built inside the cell in the form a receptor requires. And the authors were explicit about the wider methodological problem, noting that the Prussian blue staining approach had led investigators to conflate iron-rich macrophages with magnetoreceptors. Now, the uncomfortable part, and this is a hazard that runs through this entire literature, magnetic particles are in everything. Steel dissecting instruments shed them. Laboratory dust carries them. Reagents can carry them. The air in most buildings contains fine iron oxide particulates from vehicle brakes, from combustion, from industry. Working with magnetic nanoparticles at the level of a handful per cell means working at a level where the tools, the bench, and the room can contribute more magnetic material than the sample does. Kirschvink has been arguing this point for decades, and his own lab built magnetically clean facilities for exactly this reason. When his group published on magnetite in human brain tissue in the 1990s, the contamination controls were a substantial part of the work because without them, the result would have meant nothing. That's a strange and slightly demoralizing situation for a field to be in. The signature of your quarry is indistinguishable from the signature of your scalpel. Here's what I actually admire about this story, though, and it's the reason I don't read it as a failure. Look at the author lists. Kirschvink is on the 1997 paper, the 2000 paper, the 2012 paper, and the 2015 paper that undercut all of them. Winkelhofer is on both the 2012 claim and the 2015 correction. Eder found the rotating cells, and Eder was part of the team that went back and failed to find magnetite inside them. The same people who built the case took it apart. Nobody had to be dragged. They developed a sharper method, applied it to their own prior claim, got a negative result, and published it. That is what the process is supposed to look like, and it is much rarer than it should be. So where does the trout stand now? The behavioral evidence hasn't gone anywhere. Trout can be conditioned to magnetic fields. Salmonids in general show robust magnetic responses, and there's good work showing that juvenile salmon orient according to magnetic field parameters in ways that match their species' migratory routes. Nathan Putman, Ken Lohman, and colleagues have done a lot of this, and they've also shown that magnetic pulses alter the orientation of juvenile Pacific salmon, which is a result we'll come back to because of what a pulse can and cannot affect. The electrophysiological evidence from Ross v. is harder to evaluate at this distance. Recordings from that nerve-reporting magnetic responses were made, and they haven't been comprehensively replicated in the way you'd want. The anatomical evidence is where the collapse happened. The candidate cells identified in the 90s and the candidate cells identified by magnetic screening in 2012 have not survived direct examination of their contents, which leaves us with a fish that demonstrably perceives magnetic fields and a receptor that has now failed to be found twice, using two completely different search strategies by some of the best people in the field. And it brings the question into focus in a way I think is actually clarifying. Because after the beak and after the trout, the field had to confront something it had been assuming for 30 years without examining it. Finding magnetic material inside an animal doesn't mean anything at all. You have magnetite in your brain right now. That's not a metaphor, and it isn't fringe. In 1992, Kirschvink, Atsuko Kobayashi Kirschvink, and Barbara Woodford published a study in the Proceedings of the National Academy of Sciences reporting biogenic magnetite in human brain tissue. Millions of single-domain crystals per gram, with higher concentrations in the membranes surrounding the brain. The work was done under strict contamination controls precisely because the authors knew the result was worthless without them, and it's been repeated since. Human beings have magnetite in their heads, arranged in some cases in small chains, in the stable single-domain size range. Human beings also have no demonstrated magnetic sense. There have been attempts. There's a study out of Kirschvink's own group reporting changes in brainwave activity in human subjects when the ambient magnetic field was rotated in a shielded chamber. And it's an interesting result that deserves replication. And as of now, it sits well short of a demonstrated sensory ability. Nobody can find north with their eyes closed. If you could, we'd have known for several thousand years. So here's a species with millions of biogenic single-domain magnetite crystals per gram of brain tissue and no compass, which means the material can be present in the right size range, made by the body, and still be doing nothing that resembles magnetoreception. It gets worse. In 2016, Barbara Maher and colleagues published work in the same journal reporting magnetite nanoparticles in human brain tissue, with a morphology that told a story. Biogenic magnetite tends to be angular, faceted, crystallographically clean. A large fraction of the particles they found were rounded and fused, sometimes attached to other metal-bearing particles, with the shapes characteristic of material that condensed out of high-temperature combustion. Their interpretation was that a substantial amount of the magnetite in human brains came from the air, vehicle exhaust, brake wear, industrial emissions, inhaled, and, they proposed, taking a route into the brain through the olfactory system. The interpretation has been argued over, as it should be, and the argument is ongoing. I don't need the interpretation to be right for the point to land. The point is the morphology. The only reason anyone could distinguish those particles from biological ones was that biological magnetite has a specific look and combustion magnetite has a different one, and somebody had done the work of characterizing both. Now run that back through the beak and the trout. If you take an animal, dissect it with steel tools in an ordinary laboratory, in an ordinary city, stain the tissue for iron, and find magnetic particles, you have found something that could be a receptor, or could be an iron-storing macrophage, or could be a fragment off your instruments, or could be a particle the animal inhaled on a highway. Magnetic material in tissue is not evidence. It's a starting point for an investigation, and the investigation is the hard part. Which brings up the other kind of claim, the molecular kind, and the most spectacular recent example of how badly this can go. In 2015, a group led by Can Shi in Beijing published a paper in Nature Materials titled A Magnetic Protein Biocompass. The claim was extraordinary. They reported that a protein called ISCA1, which they renamed MAG, assembles with cryptochrome into a rod-shaped complex, and that this complex has a permanent magnetic moment and spontaneously aligns in magnetic fields as weak as Earth's. If true, it would be a molecular compass with no mineral involved, a receptor built entirely out of protein and iron-sulfur clusters. It was covered everywhere, and it fell apart within about a year. On grounds that should be familiar by now. The thermal floor. Run the physics on the proposed complex, even granting generous assumptions about how the iron atoms in those clusters could be magnetically coupled. The energy the complex would gain by aligning with Earth's field comes out around 10 to the minus 25 joules. The thermal energy jostling it is around 4 times 10 to the minus 21 joules. The field's grip on the molecule is tens of thousands of times weaker than the random shoving from the surrounding water. Marcus Meister at Caltech published a broader analysis in eLife, working through the physical limits of using proteins and iron-containing complexes to manipulate cells with magnetic fields, and the conclusion was the same across a whole class of claims. The energies are nowhere close. Michael Winkelhofer, Henrik Moritzen, and others wrote detailed technical critiques. Nature ran a news piece in 2017 with the headline that the compass protein had attracted a heap of criticism, which is about as gentle as that gets in print. Subsequent attempts to use the protein as a magnetic actuator in well-controlled experiments have failed. And notice what killed it: arithmetic. Arithmetic that anybody could have done before publishing, using a number that has been sitting in the middle of this field since the 1980s. 4 times 10 to the minus 21 joules. That's the bar. Every proposed magnetic receptor mechanism has to clear it. And the reason magnetite is such a durable hypothesis is that a single domain crystal chain is one of the very few structures biology can build that does. There's one more line of evidence worth putting on the table, because it's the closest anyone has come to attacking the animal problem with the tools that solved the bacterial one. In 2022, Renee Bellinger, Michael Banks, and colleagues published in PNAS an analysis arguing that homologs of magnetosome genes are conserved across all domains of life. They reported magnetite in salmon olfactory tissue in the form of compact crystal clusters, identified candidate magnetic cells, looked at which genes were differentially expressed in those cells, and found distant relatives of magnetotactic bacterial genes among them. They reported a core set of magnetosome gene homologs present in the great majority of the animal genomes they examined. That's a serious attempt. And if it holds, it would be the first real bridge between the solved system and the animal problem. The response was fast, and it was sharp. Montel and colleagues published a reply, also in PNAS, under a title that summarizes the objection completely. Magnetosome proteins belong to universal protein families involved in many cell processes. Their argument is the one you'd make if you'd been paying attention to what we walked through earlier. Remember MAM-B and MAM-M, the iron transporters? They're cation diffusion facilitators. That family exists in essentially every organism on Earth, where it mostly handles zinc and other metals and has nothing to do with magnetism. MAM-E has a protease domain, and proteases are everywhere. Several magnetosome proteins are built on GTPase folds, and GTpases run half of cell biology. So finding a distant homolog of a magnetosome gene in a salmon can mean you found a magnetite biomineralization gene. It can also mean you found a zinc transporter. The homology is real, and it's also, on its own, close to uninformative. The critics also pointed out that only a very small fraction of the genes differentially expressed in the magnetic salmon cells had any relationship to magnetosome genes at all, which makes the inference thin. Bellinger's group replied, arguing that the universal presence of the core set points toward an ancient shared origin. The exchange is unresolved and both sides are being reasonable. My own read, and it's a read rather than a verdict, is that the argument goes wrong at the level of which genes you'd want to find. The parts of the magnetosome system that make it a magnetosome system are the specific ones. The magnetochromes, that peculiar cytochrome family found nowhere outside magnetotactic bacteria. MMSE6, with its crystal templating acidic tail. MAM-J, the tether. The specific combination of MAM-K filaments positioned to hold a chain straight. Those are the parts with no generic day job. Those are the parts that would tell you something if you found them, and nobody has found them in an animal. What has been found, repeatedly, is that animals contain proteins from ancient universal families that also happen to have been recruited into magnetosome formation in bacteria, which is roughly like discovering that both a violin and a barn contain wood. If you've been keeping score, the tally so far looks bleak. 2 flagship anatomical candidates dismantled, a molecular candidate destroyed by arithmetic, a genomic argument that comes down to whether a zinc transporter counts as evidence. So it's worth being clear that the underlying phenomenon is one of the most solid things in animal behavior. It's the receptor that's missing. The sense itself is about as well established as anything in the field. Start with a loggerhead hatchling on a Florida beach. It crawls into the surf, swims out through the breakers, and heads for the North Atlantic Gyre, the enormous circular current system that will carry it around the Sargasso Sea and eventually back. If it drifts out of the gyre, it dies, either from cold water on the north side or from being carried somewhere it can't survive. It has never done this before. Nobody showed it. Its parents are gone. What Ken Lohman's lab at the University of North Carolina did, over a long series of experiments, was put hatchlings in water tanks surrounded by magnetic coils, generate the exact field parameters, inclination angle, and total intensity that exist at specific points around that gyre, and record which way the turtles swam. They swam in the direction that would keep them in the gyre. Give a hatchling the field signature from the northeastern edge of the gyre, and it swims southward. Give it the signature from the southern edge, and it turns north. Each field combination produced the heading appropriate to that geographic position, in animals that had never been anywhere and had no way to learn it. That's a magnetic map, inherited, in a reptile the size of your palm. It gets better or worse, depending on your appetite for how much these animals are doing. Earth's field drifts. The isolines that mark a given combination of intensity and inclination move over years and decades. If turtles are imprinting on the magnetic signature of their natal beach and returning to it, then as the field drifts, the beaches they choose should drift with it. Roger Brothers and Lohman went and checked that against nesting records from the Florida coast covering nearly 2 decades. Where the magnetic isolines converged, nesting density went up. Where they diverged, nesting spread out. The pattern tracked the field. Nathan Putman and colleagues ran a version of the same test on Fraser River sockeye salmon, using more than half a century of fishery records. Sockeye returning from the North Pacific have to get around Vancouver Island, and they can go north of it or south of it. Which route the bulk of the run takes varies year to year. Putman's group showed that the choice correlates with the drift of the geomagnetic field, with the fish favoring the route whose field signature best matched the one at the river mouth when they left as juveniles. Caribbean spiny lobsters, in work by Larry Bowles and Lohman published in Nature, were captured, transported between roughly 7 and 23 miles from their dens in covered containers, with steps taken to disrupt any magnetic cues they might pick up in transit, and released in unfamiliar territory. They oriented homeward, not toward a landmark because they had none, and not by retracing a route, because the route had been scrambled. They knew which way home was from a place they had never been. Bonnethead sharks, a small hammerhead species common in the Gulf of Mexico and along the southeastern United States, were tested by Brian Keller, Nathan Putman, and colleagues using magnetic displacement in a tank. Expose them to the field signature of a location well south of their capture site, and they orient northward. That result was published in Current Biology in 2021, and it was, remarkably, the first solid demonstration of a magnetic map sense in a shark, despite decades of assuming they had one. I could keep going. Eels, sea snakes, various fish, mole rats, bats, bogong moths crossing Australia at night. The behavioral literature is deep, and it's been getting steadily better controlled. Now, the physics trick I mentioned earlier, because it's the bridge between all this behavior and the question of hardware. A magnetic pulse is a very short, very strong field, oriented across the animal's normal magnetization. The point of it is selective. If an animal has permanent single-domain magnetic material in a receptor, a pulse of the right strength can flip or reorient the magnetization of those particles, and the receptor will then report something wrong until it recovers or the particles physically reorient. If the animal is using a chemical compass or an induction-based system, a brief pulse has no lasting effect at all, because there's nothing there that holds a magnetic memory. So a pulse is a test that discriminates. It asks, is there permanent magnetic material in the loop? Pulses have been applied to migratory birds, to big brown bats, to sea turtles, to spiny lobsters, to juvenile Pacific salmon. And across those experiments, orientation behavior changes. Sometimes it shifts by a consistent angle. Sometimes it becomes random. There's a pattern in the results that took years to come into focus. The pulse tends to disrupt the positional or map component of the animal's behavior while leaving the compass component more or less intact, which lines up precisely with the split we saw in birds, where the trigeminal nerve carries the map information and the visual pathway carries the compass. Then, in 2025, the turtle work delivered the cleanest version of this anyone has managed. Kayla Goforth, working with Catherine and Ken Lohman, trained loggerhead hatchlings to associate specific magnetic signatures with food. Turtles that recognize a fed field do something unmistakable. They stick their heads out, slap their flippers, sometimes spin in place. Researchers have described it as dancing, which is unscientific and also completely accurate if you watch the footage. That gives you a readout, a behavior you can score, driven by the map sense in a controlled setting. The team then attacked the two senses separately, applying an oscillating radio frequency field, the kind that interferes with radical pair chemistry, disrupted the turtles' compass orientation. It did not disrupt their ability to recognize a learned magnetic signature. Two senses, two different responses to the same manipulation, in the same animal. Follow-up work from the same group, published in the Journal of Experimental Biology later that year, went after the map sense with a magnetic pulse. The learned response weakened, and they gathered evidence pointing to the map receptors being located in the head. Put that together, and you get a conclusion that is, in my view, about as strong as an indirect argument can get. Sea turtles have two magnetic senses. One behaves like a chemical, light-dependent radical pair compass. The other behaves like a mechanical sense based on permanent magnetic material. It's used for position. It lives somewhere in the animal's head, and it can be knocked around by a magnetic pulse, which means there is, almost certainly, manufactured single-domain magnetic material sitting in a loggerhead turtle's head, coupled to a nerve right now. Nobody has ever seen it. And then, this past May, the strangest development in the whole 50-year run. A team based in Bonn and at the Max Planck Institute of Animal Behavior published a paper in Science. The first author is Clivia Lisowski, working with the immunologist Christian Kurtz, the physicist Ulf Wiedwald, and the movement ecologist Martin Wikelski. And the structure they're proposing as the pigeon's magnetic sensor is a population of iron-loaded macrophages in the liver. Give that a second, because the field spent 14 years learning to say the sentence macrophages are not magnetoreceptors, and here's a paper arguing that macrophages are magnetoreceptors. Here's what they report. Macrophages in the liver do a specific job. They break down worn-out red blood cells and recycle the iron. That leaves them stuffed with iron, packed into ferritin, and inside those ferritin stores, the iron crystallizes into oxide nanoparticles. The team characterized the magnetic properties of those cells and found them to be superparamagnetic. Millions of such cells, sitting in a liver that is well supplied with nerve endings. Then they ran the behavioral test, and the design of it is the part I want to give credit for, because it's cleverer than it first looks. They trained 34 homing pigeons on a route of about 12 miles west to east. They watched the forecast, and roughly a day before an overcast day, they gave half the birds a clodronate-based treatment that depletes macrophages. Then, under cloud, they took all the birds out and released them with tracking equipment. The untreated birds came home in about 70 minutes. The treated birds did not come home that day at all. They scattered, wandered, and only made it back after the sun reappeared. Then, the control that makes the result worth taking seriously. They ran the same experiment on clear days. On clear days, the treated birds flew home normally, straight and fast, indistinguishable from the controls. That control matters enormously. The obvious objection to any drug depletion experiment is that you've made the animal sick, and a sick bird flies badly. But a sick bird flies badly in sunshine, too. These birds were fine in sunshine. Whatever the treatment took away, it was something they only needed when the sun was hidden, which is precisely the condition under which homing pigeons are known to fall back on magnetic information. The authors proposed that the signal travels to the brain via the vagus nerve, and that detection is collective, meaning it emerges from the aggregate response of a large population of cells rather than from any single cell resolving the field on its own. Now, measure it against the specification we wrote down, because that's what a specification is for. Requirement 5, loss of function, is satisfied about as well as it can be in a bird. Remove the cells. The behavior breaks under conditions where it's needed and stays intact under conditions where it isn't. That's a real result, and I don't want to undersell it. Requirement 1 is where it gets interesting, and it's where this connects back to everything we spent the first half of this establishing. The material is superparamagnetic. Go back to the size window. Superparamagnetic means the particles are small enough that thermal energy flips their magnetization continuously, so they hold no stable direction of their own. They are, individually, magnets with no memory. That is the exact regime the bacterium spends an entire operon and a size control system making sure to stay out of, which means whatever the pigeon liver is doing, it can't be doing what the bacterium does. There's no permanent dipole to be torqued. Any detection has to come from something else. Force in a field gradient, a collective statistical alignment across many particles, some coupling nobody has fully worked out yet. The physics of that is harder than the physics of a chain of single-domain crystals, and the paper's own proposal that detection is collective is an acknowledgement of the problem. Requirement 3, that the cell be sensory and innervated, is proposed on the basis of proximity to nerves. Proximity isn't innervation. Requirement 4, a recorded magnetically modulated signal on a nerve, has not been demonstrated here. And requirement 2, mechanical coupling, hasn't been characterized at all yet. So the newest and strongest result in animal magnetoreception nails the functional test and leaves the mechanism largely open. The implication I find hardest to let go of is this one. and I'll flag clearly that it's interpretation. If this holds up, then the pigeon has no factory. There's no magnetosome island in a bird genome, no MAM genes, no magnetochromes, no dedicated compartment built to hold a crystal at a controlled size. What there is, on this account, is an ancient piece of housekeeping machinery. The recycling of dead red blood cells that happens to leave a population of cells loaded with iron oxide, and a nervous system that at some point started paying attention to what those cells were doing in a magnetic field. That's not manufacturing, that's opportunism. Evolution found a magnetic byproduct lying around in the plumbing and wired a sense onto it, which would be, if true, both less impressive and considerably more interesting than the alternative. Less impressive because there's no engineering marvel to admire. More interesting because it would mean the whole search has been looking for the wrong kind of object. 50 years of hunting for a manufactured crystal chain, when the answer might be a metabolic side effect with a nerve pointed at it. I want to be measured about this because it's one paper, it's very new, and the researchers themselves say it needs replication. Outside commentary has been a mix of respect for the rigor and open questions about whether it generalizes. Nobody knows whether songbirds have this. Nobody knows whether bats do, or sharks, or turtles. The liver of a pigeon is not obviously a template for the head of a loggerhead. And there's a physical tension between this result and the turtle result that I don't think anyone has resolved yet. And I want to put it on the record because it follows directly from what we established earlier. Magnetic pulses affect permanent single-domain material. They do essentially nothing to superparamagnetic particles because superparamagnetic particles have no remnants to disturb. The pulse hits, and a millisecond later the particles are back to flipping randomly the way they always were. Sea turtles' map sense is disrupted by a magnetic pulse. So is orientation in birds, bats, lobsters, and salmon. Superparamagnetic macrophages should not be able to explain a pulse-sensitive sense, which means either the pigeon liver system is one of several magnetic systems in these animals, or the pulse results are telling us about something in a different tissue entirely, or some part of the physics is more complicated than the clean version I've laid out. All three of those are live. None of them is comfortable. That is roughly where the field is standing at the moment. So what would actually settle it? I think it's worth spelling out because the standard here is not vague, and knowing what the finish line looks like changes how you read every announcement that comes out from now on. You would need a cell identified and located in a specific tissue in a specific animal, imaged where it sits, with its neighbors and its connections intact, using electron cryotomography on frozen tissue. Dissociating cells into a dish is how the trout candidates got lost, and everybody knows it now. That is exactly the technique that settled the question of whether a magnetosome was a free vesicle or an invagination, and it exists now, and it's improving fast. Inside that cell, you'd need magnetic material with a characterized size distribution. Narrow, sitting in the stable single-domain range for its crystal geometry. Faceted, chemically pure, with a habit that no combustion process and no dissecting tool produces. In other words, material that passes the biogenic criteria that were developed for magnetofossils and stress-tested in the argument over a Martian meteorite. The mineralogy would have to be done on the actual crystals in the actual cell at high resolution, with the contamination controls that this field learned the hard way to demand. Then you'd need the linkage, visible in the same reconstruction, a filament, a membrane attachment, some physical path from the crystal to a structure that can deform, ideally with a candidate mechanosensitive channel in the neighborhood. This is the requirement that has never been met for any animal candidate, and it's the one I'd watch for most closely, because it's the difference between a cell that stores iron and a cell that reports on a magnetic field. Then, the electrode. An afferent nerve from that tissue, recorded while the field is swept, showing a response that tracks the field parameter the animal behaviorally cares about, replicated somewhere else, by somebody else, ideally by somebody who'd be pleased to see it fail. And then the manipulation. Take that specific structure out, or disable it, and watch the specific behavior break with a control condition where the animal is fine, the way the pigeon liver study used sunny days. Meet all 5 and it's done. Nothing less closes the case, and I think the field would broadly agree with that list, even where it disagrees about everything else. What makes the continued failure so puzzling, and I don't have a comfortable explanation for it, comes down to one observation. The tools are no longer the problem. The instruments available today are better than the ones that cracked the bacterium. Cryotomography can resolve individual protein filaments inside frozen cells. Correlative microscopy lets you find one cell in a dish and then look inside that exact cell with an electron beam. Single-cell sequencing can tell you what genes are switched on in one cell at a time, so you don't need to know what the receptor looks like in order to notice that it's expressing an unusual program. Quantum magnetometry using nitrogen vacancy centers in diamond can map magnetic fields at cellular resolution, and it's already been used on pigeon inner ear tissue. Synchrotrons can do element-specific imaging on tiny samples. Somebody working on this in 1997 would look at that toolkit and assume the problem was solved within 5 years. It wasn't. And the reasons, as far as I can tell, come down to 3 things we've already walked through. The receptor is probably rare, maybe a few hundred cells in an entire body. It has no known marker, so you can't sort for it, stain for it, or label it. And its defining physical property, magnetism, is shared by contaminating dust, by immune cells doing housekeeping, and by particles the animal breathed in on the way to the lab. There's a 4th reason, and it's the one that unsettles me most, which is the possibility that the search has been shaped by a wrong assumption. Every anatomical hunt in this field has been implicitly looking for something that resembles a magnetosome. A crystal, made on purpose, held in place, wired up. That expectation came from the bacterium, and the bacterium is the only fully worked example anyone has, so of course it shaped the search. If the pigeon liver result holds, the object being looked for all along was a pile of recycled iron in an immune cell, doing something collective and statistical in an organ nobody was examining because livers are not where you keep your senses. There's a practical edge to this too, and I'll keep it brief because it doesn't need dressing up. Loggerhead turtles, salmon, and sharks are navigating on a magnetic signal in an ocean that we are steadily filling with electrical infrastructure, undersea power cables, offshore wind installations, seabed survey equipment. Whether any of that matters to a migrating animal depends on what its receptor is and how it responds, and right now we can't answer that because we don't know what the receptor is. You can't predict how a sensor fails when you can't identify the sensor. There's something I'd honestly like to hear from you here, and I mean it as a real question, not a rhetorical one. If somebody handed you a freshly euthanized loggerhead, a cryo-electron microscope, and 1 week, where would you cut first? The ophthalmic branch of the trigeminal is the conventional answer, and it's conventional for good reasons. But the pigeon result just made the liver a serious proposition, and the inner ear has been a candidate since the Lagina work. And there's a real argument that nobody has looked properly at the olfactory system in a reptile. Put your answer in the comments. Name the tissue and say why. I go through them, and the arguments people make about where a sense ought to live are frequently better than the ones I've seen in review articles because people who aren't inside a field aren't carrying its assumptions. If you've got a background in materials science or imaging, or you've spent time on a boat watching animals do things that shouldn't be possible, I especially want to hear from you. And if you want more of this kind of thing, the slow kind, where we stay with a problem long enough to see the shape of what's missing. Subscribing is how you get it, and dropping a like on the way out tells me this is the register worth staying in. It's a small thing, and it makes a difference on my end. Now let me say the gap as precisely as I can, because that's the whole point of everything we just went through. For one organism, we have the complete answer. In magnetotactic bacteria, we can name the genes, roughly 30 of them, sitting together on an unstable island in the chromosome. We can describe how the membrane compartment forms and which proteins are required for it. We can name the transporters that carry the iron across the compartment wall and the redox proteins that hold the iron in a mixed oxidation state so the mineral comes out as magnetite. We can point to the protein that templates the crystal and the operon that sets its size, and explain in physical terms why the size has to fall between roughly 30 and 120 nanometers, and what fails at each end. We can name the filament that holds the chain straight and the protein that clips the crystals to it, and we can predict what happens when you delete that protein, and we have deleted it, and the prediction was right. We can take those genes out of that organism, put them into a bacterium that has never made a magnetic crystal, and watch it start manufacturing compasses. That is the level of understanding available for one class of single-celled organisms living in the mud of ponds and coastal sediments. For every animal that navigates the ocean by the geomagnetic field, and there are many, and the behavioral evidence for them is excellent, we cannot point to one cell, not one, in any species. We know the sense exists because we can measure it, condition animals to it, and break it with a magnetic pulse. We know approximately which nerves carry it in a couple of species. We have a strong new claim about a cell type in one bird, with a functional test behind it and a mechanism still to be worked out. What we have never done is put a piece of tissue under a microscope, find the structure, characterize the material, trace the wire, and say, there it is. That's the receptor. That's what it's made of. That's how it works. 51 years since the bacteria were properly described, 63 since Bellini watched them in Pavia, and nobody listened. One organism where the manufacture of a biological compass is understood down to the gene. 0 animals where anyone can identify the cell that carries one. Everything happening in this field is happening in the distance between those two sentences.